Treats and hats. Ready for us? And I'm going to pass it right over to Jay.
All right. In an era of escalating dissertation exits our story phenomena, we have hats, all sorts of snacks, try to identify what snack goes with what part of Karolina's talk. But it's my pleasure to introduce Karolina Zabinski, I should say, Dr. Karolina. And Karolina was from the COVID cohort. Those of you in the COVID cohort know how tough it was to try to figure out how to navigate becoming a grad student in a place where the whole reason why you came to grad school doesn't exist, being with people and learning from all sorts of one-on-one interactions with them. But she and her whole cohort, as we have heard many of this quarter, were incredibly successful. And the thing that I want to sort of just sort of emphasize about Karolina is that I can remember when she applied here, one of the things that her letter writers pointed out were like, she's just interested in everything. And they meant that both as a compliment and you gotta watch out. She's just as excited about everything. And I think anybody who knows Karolina knows that she is actually pretty excited about everything. And what you're gonna see in this dissertation, I think, is how Karolina sort of actually harnesses that interest in all sorts of different things and is really good at going and finding what she needs from people who are not her advisor, because her advisor knows nothing. Maybe a little bit about a few things, but most of what she's going to talk about is stuff that, she went and talked to Gina or Jonathan or found someone from Scripps or talked to somebody about genetics. So she really kind of drew together a lot of different things from field ecology, sort of microbiome research, and a whole bunch of chemistry that you're not going to see, but she'll be working on in her postdoc. And I'm not going to tell you about that because I will leave that to Karolina. But she is now officially Dr. Karolina Zabinski. The paper has been signed. And I'm really excited to let her share her research. So Karolina, take it away.
So, do I first? Yeah. 1 second. This way. Yep, there you go. Oops. All right, thank you so much for that introduction, Jay. I'm really excited to be sharing some of the work that I've gotten a chance to conduct here at Davis over the past 6 years. So I'm really interested in understanding what are the ecological and evolutionary processes that drive species persistence in a changing world. And so I gotta do it. I gotta shout out my because I really think we embody that persistence in a changing world. And we've almost all graduated and we're here, yeah. So typically, when we want to understand how species will persist in changing environments, we can compare how individuals vary in their resilience to environmental stressors. So when we compare individuals and environmental stress, we think about these intrinsic differences amongst individuals like genetic effects. But we can also compare non-genetic differences across individuals like microbial symbioses. So here, each clump of microbes that's a distinct color represents an individual's unique microbiome, which for the purposes of this talk, I'm defining as a group of bacterial partners. So all individuals on the planet are associated with bacterial partners or microbial symbionts, which come together to form an individual's microbiome. And that microbiome can impact an individual's ability to respond to a changing environment. And that's because I like thinking about bacteria as these little Ziploc baggies of genes and chemicals, which can correspond to changing a host's genomic and metabolic potential. So now, when we build our framework of species persistence in a changing world, we have all of our individuals or hosts of interest interacting with their microbial symbionts in changing environments, which come together to drive these ecological and evolutionary processes. So it was wanting to understand how these intrinsic genetic differences amongst individuals may be coupled with these non-genetic differences across individuals, thinking about their microbial symbionts, which really drove a lot of my dissertation's research, trying to understand how microbes contribute to ecological and evolutionary processes within a species. Now, I'm a marine ecologist, so when I think about a changing world, I'm really interested in the increasing frequency, duration, and magnitude of extreme climate events. So specifically thinking about marine heat waves and temperature stress. So that's our framework that we're going to be thinking about today. So that brings us to our study system that we'll be focusing on here, which is the species of seagrass, eelgrass, or Zostera marina. By the way, if at any point y'all want to take more mud pies, please do so. They've been made with love. Yeah. So eelgrasses are found up and down the northern hemisphere coast in shallow subtidal and intertidal mudflats. And I like thinking about eelgrasses as forming the nursery habitats of our oceans. So they're these marine habitat forming or foundation species. And to understand how microbial symbiosis is impacting these plant health, we have to learn a little bit about the biology of these plants first. So kind of sharing the story of microbial symbiosis of these plants. So first, eelgrasses can reproduce sexually. So they are a marine flowering angiosperm, but they can also reproduce clonally, which means that one individual or one genet can have multiple generations within that same plant. So what this means is that the legacy of stress or any lasting phenotypic response of an organism in response to a stressor can be passed down across clonal generations. So if we think about what traits might be important in thinking about in terms of temperature stress resilience across generations, one trait that we can think about is that clonal offspring size, where larger clonal offspring can be indicative of more stress resilience because their clonal offspring are larger. But we can also think about traits below ground. So here in the rhizome organ of the plant is that storage organ of the plant that also provisions our clonal offspring. So if we have larger rhizomes or that are heavier with more biomass, that can be an indicator of stress resilience because there are more provisions available for our clonal offspring. But if our rhizomes are getting longer or they're elongating, that could actually be an indicator of stress response where our plants are trying to navigate or escape a heterogeneous landscape or escape from a stressor. So we've talked a little bit about the biology. The second thing that I want y'all to take away is that even though all of these plants look like the same shade of green, there's actually a ton of genotypic and phenotypic diversity amongst individuals, both within a population and between populations. So among a population, individuals vary in their clonal offspring and below ground traits in response to temperature stress. And across populations, populations exhibit local adaptation and reciprocal field transplants, and they also exhibit high genomic structure in response to temperature. So what about the microbes? Where do the microbes come into play? So water. Okay. Now, again, as a reminder, each clump of microbes that's a unique color represents a distinct microbiome. And we're starting to gain an understanding, we're finding that eelgrasses do assemble distinct root and leaf microbial communities from their surrounding water column and sediment. And we're starting to gain an additional understanding that these microbial partners or these microbiomes can impact overall plant health. So let's close our eyes and dig below ground. So below ground, sulfur oxidizing bacteria, which I love that they're abbreviated as SOBs throughout the literature, are the most ubiquitous symbiont below ground throughout the world in eelgrasses. And this makes sense because these plants inhabit low oxygen, sulfidic rich environments. And hydrogen sulfide is actually a phytotoxin to these plants that shuts the photosystem down for these plants, eventually leading to plant death. So the way that this relationship works between these plants and these sulfur oxidizing bacteria is that the plants release photosynthetically derived oxygen and carbon into the root system below ground as food for the bacteria. And then these sulfur oxidizing bacteria detoxify the sediment, converting the hydrogen, core water hydrogen sulfide into non-toxic forms of sulfur like sulfate. So overall, these plants are happy. But heat stress can actually destabilize this symbiosis, where under heat stress, these plants have less photosynthetically derived oxygen and carbon available to feed their sulfur oxidizing bacteria. And actually, these sulfur oxidizers become replaced by sulfate reducing bacteria that reduce that sulfate into hydrogen sulfide, increasing the toxins below ground that eventually kills the plant. So these sulfur cycling bacteria really distinguish the eelgrass below ground microbial symbioses throughout the world, or from the surrounding sediment. But what I didn't tell you is that the majority of root microbial symbionts are these other taxa that we don't know the actual function of, and it's these taxa that really distinguish individuals from each other as well as populations. So interactions among root symbionts may be impacting the eelgrass ecological and evolutionary processes. Now, thinking above ground, eelgrass leaf microbial symbionts are also distinct from the surrounding water column as well as leaf mimics. So we're starting to gain an appreciation that leaves might actually be assembling their phylospheric microbiomes. And even though we know less about leaf microbial symbioses, we do know that heat stress impacts both leaf physiology and microbial symbionts. So in non-stressful conditions, the plants have low oxidative stress, so they're not producing a lot of reactive oxygen species, and they have low amounts of pheophytin levels. And pheophytin is a degraded form of chlorophyll. They're also experiencing low wasting disease severity. If you want to learn more about wasting disease, talk to my lab mate, Serena. And they also have high microbial diversity. Now, under heat stress, these plants are actually experiencing high oxidative stress physiologically, and they're producing more pheophytin levels, which are those degraded forms of chlorophyll, and they're experiencing higher disease severity. And at the microbial level, their microbiome is less diverse, and this is coupled with an increase in abundance of these bacteria called saprophytes, which are taxa that are known to degrade leaf tissue. But overall, when we think about these leaf bacterial changes, we don't really know whether these changes in leaf microbial symbionts are beneficial or detrimental to their hosts. So together, it's using this framework, uniting seagrass within and among population variation under changing environments, that unite with variation in their microbial symbionts by characterizing their microbes using 16S rRNA sequencing of the V3, V4 region, that I use to understand how microbes contribute to ecological and evolutionary processes within eelgrass. And so I do this by first assessing how microbes may be contributing to individual variation and heat stress resilience, then by trying to understand how microbes may be contributing to local adaptation between populations, and then finally, by understanding the extent to which legacy effects may be differentiating plant traits versus microbial symbionts between populations. Okay, so in my first chapter, just as a note, I'll always have my collaborator, like my co-authors, pictured up on the slides. And then I always tell my undergrads that the only I in science is in the word. I'm looking at my undergrads right now. The only I in science is in the word. So I have all of these other people that help put these projects together just written out, because I didn't have enough space for everyone. Yeah, so the other thing is that whenever Jay gives talks, he always puts embarrassing pictures of me and of the lab on the slides. And today we're going to talk about 3 projects. So there's 3 opportunities. Pictures of my advisor. So I call this the eelgrass angel. With me on my shoulder in the field. WWJD, what would Jay do? Yes. In my first chapter, I'll be sharing a bit about this project, trying to understand how do microbes contribute to individual variation in heat resilience. And so this work was really inspired by some coral folks who found that after a marine heat wave, there are some corals like this Passalacra who had remarkably stable microbiomes throughout that marine heat wave. But there was this other coral, Acropora, that had highly restructured microbial symbionts after that marine heat wave. And so they couldn't tell whether these patterns were beneficial or detrimental to their hosts, but they coined this term of microbial restructuring as microbial flexibility. And they hypothesized that there could be 2 patterns of microbial flexibility in these species. So they first denoted this Passalacra as a microbiome regulator because it had very low microbial flexibility across this stressor. And then they denoted that Acropora as a microbiome conformer because it had really high microbial flexibility. So I study one species, just the eelgrass, there's a lot going on in eelgrass, okay? Yeah, so I was interested in understanding when and where is microbial flexibility beneficial within a species. So first, let's think about these patterns of microbial flexibility, more specifically this regulator pattern where we have low microbial flexibility or that low microbial restructuring. So we can start visualizing this pattern of flexibility in NMDS space, where an NMDS plot is just a two-dimensional way that we can visualize multidimensional data. So each point here represents a microbiome, where points that are closer together are more similar to each other, while points that are further apart are microbiomes that are more dissimilar to each other. So here, these open circles represent individuals that are not stressed and will never experience that stressor. And that red triangle denotes the not stressed individuals as average microbial communities, or the not stressed individuals as centroid, okay? So now these black circles represent the individuals which will one day experience stress before the stressor. They're microbial communities. And these dashed lines are just us measuring that dissimilarity between each stressed individual and that average microbial community in non-stressed conditions. So what we see is that before the stressor occurs, both of our treatments look pretty similar to each other. And with the regulator pattern of flexibility, these stressed individuals still remain pretty close to the average microbial community under non-stressed conditions. So there's low microbial restructuring. And that pattern of flexibility can be associated with both high and low host performance, where high performers may be maintaining their essential symbionts, while low performers may not be acquiring symbionts with high acclimatization potential. So this is the regulator pattern of microbial flexibility. So now let's pivot to the conformer pattern. So conformers here, after the stressor event, experience higher microbial restructuring, so higher microbial flexibility. And this pattern of flexibility can also be associated with both high and low host performance, where here high performers may be maximizing microbially mediated acclimatization, while low performers may be losing their essential symbionts or increasing opportunities for pathogens to arise. So overall, if we look at this state space of host performance versus microbial flexibility, these if we see intraspecific variation in microbial flexibility, that could indicate that there could be differences in microbial functions, differences in host control of their microbial communities, or differences across individuals in their parallel changes between themselves as well as their microbial symbionts. Okay, so the way we went about testing this is we took 7 unique eelgrass genets, or unique genotype individuals, and we took replicates for each of these genets, and we exposed half of these individuals to ambient conditions for 15 weeks, and we exposed the other half of individuals to a simulated marine heat wave event, where we held them for ambient temperatures at 5 weeks, elevated those temperatures for 5 weeks, and then ramped the temperatures back down for 5 weeks. And because I was interested in seeing when microbial restructuring may be associated with host stress resilience, we measured for every individual's leaf and root microbial communities before we exposed them to that heat wave. So right after that ambient period in that first gold star, and then after 5 weeks of recovery to that marine heat wave period. So where the second gold star is. So looking at some host data, the y-axis here shows the difference in the number of clonal offspring produced for every warmed individual relative to the average number of clonal offspring produced across all of our genets, which is the x-axis. And so what we see is that more resilient genets had about a 0% reduction in the number of clonal offspring produced between treatments, while less resilient genets had up to a 44% reduction in shoots compared to their control counterparts. So what I just want you to take away from this graph is that there are more heat resilient genets, and there's also less heat resilient genets on this continuous scale. So right now what we've done is we've calculated our y-axis, which is our difference in shoot production, or our metric of host stress resilience. And now it's time to calculate our x-axis, or microbial flexibility. So the way that we do this is for each genet, we construct our reference microbial community, or that average microbial community composition in a control condition, so our red triangles. And then what we do is we measure that average dissimilarity between every warmed individual to the average microbial community under control conditions. And we repeat these calculations across all of our genets in the ambient and recovery time periods, such that now we have our average microbial community dissimilarities between our warm samples and our reference communities. And now we can just measure microbial flexibility as the difference in that dissimilarity, which captures that microbial restructuring. So now we have our host performance, our microbial flexibility, and we can run a linear regression to just associate our two variables together. So first, we're going to look at some root data. So our difference in shoot production is on the y-axis, and we have root microbial flexibility on our x-axis. So what we're seeing here is that resilience negatively associates with root microbial flexibility. So in other words, our more resilient genets are exhibiting a regulator type pattern of microbial flexibility, while our less resilient genets are exhibiting a conformer type pattern of flexibility. So overlaying this data onto our state space, we get something like this. And now if we're going to pivot above ground to our leaves, what's interesting is we see this opposite pattern where resilience positively associates with leaf microbial flexibility, where now our less resilient genets are exhibiting a regulator type pattern of flexibility, while more resilient genets are exhibiting a conformer type pattern of flexibility. So overall in our state space, now our leaves are encompassing opposite. I'm not even going to say the quadrants of our state space. So overall, we're going to fill in all the blanks here. And what's really interesting is that all of these patterns that we observe in our microbial communities between roots and leaves and across genets are not driven by a few differentially abundant taxa that distinguish genets or strategies of microbial flexibility or even temperature treatments. But rather, genets differ across the board in their microbial communities. So if we have these more and less resilient genets, just from the get-go, genets start off with different microbial communities across the board. And that could be differences in taxa, but that could also be differences in the relative abundance across the board of all of their taxa, even if they share the same taxa. Such that after the stress, the microbial communities have restructured across the board again in a genet-specific manner. So there are no specific taxa differentiating treatments, genets, or patterns of microbial flexibility. So altogether, what we see is that our more resilient genets are exhibiting a regulator pattern of flexibility below ground and a conformer pattern of flexibility above ground. And again, when we look at this state space, we cannot specifically disentangle whether these changes are caused by microbial functional changes, host functional changes, or differences in parallel changes between our hosts and our microbial symbionts, but we can start to hypothesize why there might be differential strategies or what might be the causes of these differential strategies or patterns. Sorry, not strategies. I don't want to get y'all patterns. Okay, so we found that regulating roots may be beneficial under stress, and this could have been stemmed from having just across the board, genets microbial communities might have differed community-wide that could have resulted in differential functional expression that led to different processes that are critical for plant health. So just as an example, more resilient genets could have been maintaining processes important for sulfur cycling like oxidation, while our less resilient genets may not have been able to maintain those critical microbial functions. There could have also been differences in host control, where our more resilient genets may have maintained greater host control, while our less resilient genets may have not been able to maintain host control. And finally, there could have just been differences in those parallel changes between our hosts and our microbial symbionts, where more resilient genets, their root morphologies or something about the way that their roots were growing led to a more stable sediment environment. But in our less resilient genets, the way that their root morphologies were growing across our temperature stress, that led to a less stable sediment environment. Now above ground, we found this opposite pattern where conforming leaves was associated with higher stress resilience, which may have stemmed from, again, these microbial community changes or overall functional changes that resulted in differences across genets related to the processing of those pheophytin or reactive oxygen species between more and less resilient individuals, or differences in host control, as well as differences in the extent to which the genets and their microbial communities changed, where our more resilient genets may have had higher leaf turnover rates, leading to more flexible leaf microbial communities, while our less resilient genets may have had lower leaf turnover rates, leading to lower microbial restructuring above ground. But altogether, what this experiment is showing us is that there are interactions among plant genets and heat stress, which are coupled with genet variation in microbial community flexibility, or the extent to which genets microbial communities are restructuring. which together may be contributing to interspecific variation and stress resilience. But more broadly, what these results are showing us is that microbial dynamics, rather than key taxa, may be greater indicators of host health under environmental stress, serving as this great springboard for future work exploring the mechanisms or the functional consequences of microbial flexibility. Okay, here we go. So for my second chapter, I was interested in understanding how microbes may be contributing to local adaptation between populations. And so this work really relates to work done on the human gut microbiome, where individuals from populations where seaweed is present are able to process those complex carbs associated with seaweed, but individuals from populations without seaweed present are unable to process those complex seaweed associated carbohydrates. And what scientists are finding is that this is because there are interactions between those seaweed-associated bacteria and the human gut microbiome that enable people from populations along the coast to process those complex seaweed-associated carbs, showing us how microbes may be contributing to processes like home site advantage. So I was really interested in applying this concept into marine systems to understand how among microbe interactions may be differentiating eelgrass populations. And so that brings us to these two populations of eelgrass located in Tomales Bay, and they're only about 12 kilometers apart from each other, but they actually experience really different environmental conditions from each other. So first, one of the characteristics that differs is the population denoted by the black star towards the mouth of Tomales Bay experiences sand-dominated sediment, while the population towards the back of Tomales Bay experiences this finer grain size or clay-dominated sediment. So you can think like Oreos up here, pudding down here. But they also experience really different temperature profiles. So our sand-dominated site experiences cooler temperatures throughout the year, and our clay-dominated site experiences hotter temperatures towards the back of the bay, particularly during those summer months. And so in a reciprocal field transplant, what we found is that they both exhibit local adaptation. So we wanted to isolate which of these environmental factors are contributing to home site advantage. So first, when isolating the impacts of temperature and home site advantage between these two populations, what we find is that the plants from the sand-dominated site grow worse at temperatures related to the clay-dominated site, for those higher temperatures, but plants from the clay-dominated site grow equally well at both temperature regimes. So when I joined the lab back in 2020, I was really interested in understanding how does sediment drive home site advantage between these two populations and how the microbiome may be playing a role in these interactions. And so to do this, we conducted a fully processed common garden experiment where we collected sediment from each site and we brought it back to the lab. And so we filtered out any end fauna, which are those sour gummy worms. Okay? And then we collected plants from both sites. We rinsed off any adhering rhizosphere sediment, and we either planted those plants in their home or foreign sediment type. And we grew the plants in their home or foreign sediment type for 15 weeks. And afterwards, we measured plant functional traits as well as for their microbial communities. And I'm going to pause before we get into some data because I've been really enjoying this trend through PopBio on exit seminars of tallying all of the Herculean efforts that they've done. So we've had hundreds of hours of like video footage processed, tens of thousands of amphipods tethered to string, millions of DNA base pairs, like processed and single cell RNA ran, like that's above my brain. This is amazing. So I was like, what can I contribute to my cohort? What can I do for my cohort? And I'm like, I'm strong. So throughout my PhD, with the help of my lab and my wonderful assistant, Anna, who is a collegiate athlete, so I think that's one of the reasons why we did so well, is we collected 10 agricultural sized tomato bins worth of waterlogged marine sediment, which I did some hand wavy math with one of my engineering friends to calculate how much weight that encompasses. So we had 10 agricultural sized tomato bins. For each of those bins, we had to collect it from the field for being conservative. We collect it from the ocean, we pot it, and we unpot it. That's not even counting lifting the plants from the tanks for measurements, moving them around for acclimation, none of that. That's it, 3 times. And that equates overall to 55,781.25 pounds or 27.9 tons of sediment moved throughout my PhD with Anna and my lab mates and random people walking along the beach, looking at me and Anna going, "Do you need help?" So if you're a more visual learner, that equates to 4 African elephants. So if you want workout regimens, and if you want to practice, like in 2 weeks, we'll be doing this in the field. So
Come on
so,
over.
yeah, so when they say like you
Tom
move,
Sawyer.
yeah, so when they say you move mountains during your PhD, this is my mountain. So I took a picture of all the sediment afterwards. So Nikki endearingly calls this Stack Mountain, but maybe we can call it, we can like put our little names like of every person who's dumped sediment back of Bodega Parade Lab. Yeah, so that's my mountain. Yeah. Okay, so now let's pivot to science. So we're first going to look at some plant functional traits. So on the left-hand side, we're going to look at rhizome length, which is our stress response of the plants trying to escape a toxic environment. And on the right-hand side, we're looking at pore water sulfide, which is our phytotoxin of the plants. So our x-axis, we have our sediment type that the plants were grown in, and the colors of the points denote what population the plants are from. So first, when looking at rhizome length, what I want you to take away from this graph is that plants from the sand-dominated site grow equally well in both sediment types. But plants from the clay-dominated site have an increased rhizome length through that stress response when grown in their foreign sediment type. Now, when we look at pore water sulfide, we see a similar pattern where plants from the sand-dominated site have equally low pore water sulfide concentrations, which are at non-toxic levels for the plants. But plants from the clay-dominated site have an increased pore water sulfide in that opposite, in their foreign set when grown in their foreign sediment type. And these levels of pore water sulfide are toxic to eelgrass. So in all, what's happening is that only the clay-dominated plants are exhibiting a stress response and experiencing a stressful environment. So now when pivoting to our microbial communities, what we're going to look at is the effects of population versus sediment type on microbial community composition. First, looking at just our sulfur cycling taxa, which make up about 1/3 of all of our individuals' microbiomes. And then we're going to look at all of those other taxa, which make up about 2/3 of all bacteria in our microbial communities. So when looking at just sulfur cyclers only, what we see is that only sediment drives sulfur cycler composition. And this is driven by there being more reducers in our clay-dominated sediment, which makes sense. We would think that in finer grain sizes, they're more anoxic, more sulfide rich, more sulfur reducers. So, and then in our sand-dominated sediment, we find there being more sulfur oxidizers, which again makes sense. Larger grain size, more oxygen, more sulfur oxidizers. But then if we look at all of the other taxa, what we find is that both population and sediment origin drive microbial community composition in those taxa that make up 2/3 of the overall microbial communities. So overall, both populations have the same amount of sulfur cycling bacteria in each sediment type. So first, in that sandy sediment, there were more sulfur oxidizing bacteria, which we would expect that to be associated with lower levels of pore water sulfide and healthier, happier plants. And in our clay-dominated sediment, we had more sulfur-reducing bacteria, which we would expect to be associated with more stressed plants and higher levels of pore water sulfide. That's actually not what we see in our experiment. So instead, when we expose each population to either its home or foreign sediment type, what we find is that plants from the sand-dominated site grow equally well in both sediments, but plants from the clay-dominated site grow worse in the sand-dominated sediment. So considering that both population and sediment origin drove that microbial community composition in those other taxa, which make up about 2/3 or the majority of microbial community composition, what I think might be going on is that there may be some interactions among the sulfur cycling bacteria and those other taxa, in addition to the plants under these changing sediment environments that are all contributing to homestead advantage between these two populations. Or in other words, plant soil feedbacks. The land plant people already know about this, but that's what I think might be going on in these marine systems. So when we think more broadly and place our results within the broader concept of home site advantage of temperature, what we find is that our results of sediment are in the opposite direction as our home site advantage in temperature. And so even though we don't know the exact mechanisms as to how we're seeing this opposite home site advantage between temperature and sediment, together what I think that my experiment is contributing is that there may be some trade-off in the plants between responding to temperature versus assembling the appropriate microbial communities across different sediments, together explaining how multiple environmental factors can simultaneously differentiate populations and how microbes may be contributing to those patterns. Okay, so the last project here. We've had this since July of 2020. So the extent to which, like, by an anonymous artist, okay, the extent to which legacy, so here we'll talk about the extent to which legacy effects differentiate plant traits versus microbial symbionts between populations. So for this last example, we're going to focus on these clonally reproducing organisms like strawberries, where a lot of this work has been focused on, or seagrasses. So legacy effects are any lasting response of an organism's performance to a given condition across generations, or in other words, if the parental treatment impacts offspring performance. So here, if we have this F0 generation and we expose them to either ambient or stressed conditions, in the next generation, if we also expose the F1 or the offspring to either ambient or stress conditions, if the parental generation's condition matters to offspring performance, we call this a legacy effect. And so legacy effects can be particularly impactful for clonally reproducing organisms like our seagrasses, as clonal generations may be lacking the genetic variation that comes from recombination associated with sexual reproduction. So there are two types of legacy effects that we'll be talking about today. The first is a beneficial legacy effect, which happens when the F0 generation is so stress resilient that it has enough reserves left over to carry over to that next generation, so that the offspring generation performs equally well, regardless of what condition it is experiencing. And the second legacy effect that we'll be talking about today is a detrimental legacy effect, where the F0 generation is so susceptible to stress that it uses up all of its reserves, such that the offspring generation doesn't have enough reserves left over from its parental generation, and it just has lower performance regardless of the condition that it experiences. So we're starting to gain an appreciation in the literature that legacy effects can be context dependent. This is from a CPB alumna, Isabel Nyland. So, or that the historical exposure of a population could mitigate the direction and magnitude of a legacy effect. So we would expect that detrimental legacy effects will occur in populations that are rarely exposed to a stressor, and beneficial legacy effects we expect to occur in populations that are frequently exposed to a stressor. So considering that plant microbial communities are assembled by rhizome provisions, which also provision offspring size, and both of these are impacted by legacy effects, I was really interested in understanding how legacy effects may be differentiating populations, thinking about their plant trait responses as well as their microbial communities. And so to do this, we have to go back to our two populations we talked about earlier, but this time, instead of focusing on each environmental factor separately, we're going to focus on their exposure history to temperature stress, okay? So here, the population towards the mouth of Tamaulipas Bay, I'm denoting with the purple star, and the population towards the back of Tamaulipas Bay, I'm denoting with a gold star. So this graph shows us some temperature logger data from the field over the past 7 years, counting the number of days each population experiences water temperatures greater than 21.5 degrees C, or that temperature that induces that one-way home site advantage between the two populations. And so what we're finding is that that purple population towards the mouth of Tamaulipas Bay rarely, or comparatively rarely, experiences those higher temperatures relative to that frequently, or the gold population. So moving forward, I'll always denote the purple population as our rarely exposed site and that gold population as our frequently exposed site. So how did we test for these legacy effects? So we went out into the field and we collected 200 adult F0 plants from each site, and we brought them back into the lab and planted them in some unvegetated outgroup populations sand from Bodega Harbor to just cut out any possible homestead advantage of sand. So after that, we exposed half of those F0 individuals to either ambient or the other half to stress conditions of 5 degrees C above ambient, and we grew those plants over the summer. Afterwards, after that phase of the experiment, we allowed the plants to overwinterize, and then the following summer, we collected 2 F1 offspring from each F0 parent plant. We cleaned off the roots of any adhering rhizosphere sediment for each of those offspring, and then we planted each offspring in unvegetated sand again. Afterwards, we exposed those offspring to either the same or opposite condition that their parental generation shoot was exposed to, and we also, in all the tanks, had some unvegetated sand control pots to measure for microbial community assembly. So after this phase of the experiment, we measured the plant functional traits of the F1 generation, and we also measured for root and rhizosphere microbial community composition in our F1 generation, along with our sediment control pots. And so it's using data from this phase of the experiment that will identify the extent to which legacy effects differentiate plant versus microbial responses to heat stress. So first we'll look at the size of our clonal offspring produced in that F1 generation first. So that's boxed in our upper left-hand panel. And so we'll first look at our responses when the F1 generation was under ambient temperatures, which is our left-hand facet, and then we'll look at these responses when the F1 generation was stressed, which is our right-hand facet. Our x-axes represent our population origin, where the plants are either from the rarely exposed or frequently exposed site, and the colors of the bars denote the F0 or the parental treatment, where blue bars represent that the parental generation was under ambient conditions, and red bars denote that the parental generation was under stress conditions. So the asterisk above the coupled bars denotes that there's a significant difference in the effect of parental treatment on offspring performance between the two populations. So what this means is that in the rarely exposed population, when the F1 generation is held in ambient conditions, if the parental generation was warmed, their clonal offspring are smaller. But for our frequently exposed population, when the F1 generation was under control conditions, if their parental generation was warmed, their clonal offspring are larger. Now, if we look at the F1 generation under stress conditions, we see the same pattern where for our rarely exposed population, if the parental generation was warmed, the clonal offspring are smaller. And for our frequently exposed population, if the parental generation was warmed, our clonal offspring are larger. So overall, what this is showing us is that our rarely exposed population is exhibiting a detrimental legacy effect, while our frequently exposed population is exhibiting a beneficial legacy effect. And sure enough, when we look at these legacy effects across a suite of plant functional traits, what we find is that the rarely exposed population consistently experiences these detrimental legacy effects, while the frequently exposed population experiences these beneficial legacy effects. Now we'll pivot to the microbial communities, where here we have our rarely exposed populations microbes on the top row, our frequently exposed populations microbes on the bottom row, and each facet represents the offspring treatment of either in ambient or stress conditions, and the colors of the points denote the parental treatment of them being in ambient or stressed conditions. And we can breeze past this because what this data is showing us, it might look like a cloud to you, and that's true because there's no effect of parental treatment on offspring microbial assembly. And we see this in the roots and the rhizosphere. But upon further examination, what I'm finding is that in these unvegetated sand controls, they have lower microbial diversity or a more depauperate microbial community. And that depauperate microbial community is consistent with where they look really similar to our root and our rhizosphere microbiome. So what I'm finding is that we did a really good job cleaning the plants and using a depauperate sand community. And I think that might have prevented any intraspecific variation in legacy effects from being observed in this experiment. So overall, at the host level, the fact that we found legacy effects for the same plant traits but in opposite directions is further supporting that historical exposure to stressors may determine the context and direction of legacy effects. And in our microbiome, we actually don't see these legacy effects occur in root or rhizosphere microbial assembly. But what I want you to take a moment is to recall that from my last chapter, it's when we expose these plants to that rich, organic, different types of grain sizes that we start seeing these among microbe interactions with their hosts and that variation to start seeing where microbes might be contributing to host site advantage. So considering in this chapter we use the same populations, I think there's still the potential for legacy effects to be occurring under natural selections if we incorporate these two different sediment types into this system. So to recap, throughout my PhD, I've been interested in understanding how microbes contribute to greater ecological and evolutionary processes within a species. And so in my first chapter, what I found is that regulating roots while conforming leaves may be beneficial to seagrasses under heat stress. But more broadly, what this is showing us is that microbial dynamics, rather than key taxa, may serve as greater indicators understanding host health under changing environments. In my second chapter, what I found is that sediment drives opposite home site advantages temperature, which is coupled with population-specific microbial changes. So what this could be telling us is that there could be plant microbe environment interactions that are mediating population differentiation to multiple environmental factors. And finally, in my third chapter, what I find is that stress legacy effects differentiate plant traits, but not microbiome responses. And so what my chapter is telling us is that historical exposure may mediate the context and direction of legacy effects. But when we think about our microbes, a greater microbial pool may be needed to initiate those legacy effects below ground. So when we have this intraspecific variation in our hosts and this variation in our microbial symbionts under changing environments, I was drafting and I was like, let's start putting together some pieces. And I was like, bam. And I was like, oh my gosh, that is so much stuff is going on. And I was like, wow, species persistence depends on so much, but that's just scratching the surface. That's only 3 projects. What am I doing? And it's a good thing. Judy Bronstein, I love her because I met with her and she was like, well, that's why careers are long. Like, it's a good thing careers are long. And Anya says that too when we talk. So it's really been a privilege to work at UC Davis. And I'm really thankful for the CPB community because I've not only learned how to build this framework of hosts, microbes, and the environment, but I've also learned how to do ecology and learned how to do some evolution. Yeah, I didn't want to overstep. Yeah. But I'm just learning how to be a scientist. And so I'm really thankful for having come to Davis for my PhD because I'm excited to go forward and I'm excited to dig deeper into those mechanisms of microbial symbioses, the consequences of symbioses, and I'm excited to stretch myself to incorporate new species into this framework. And so I'm really excited to announce that I got the Moore Foundation Fellowship for UC Santa Cruz. Yeah. To explore how microbes may be contributing to eco-evo processes between species under the guidance of Drs. Christy Croker, Ingrid Parker, and Kathleen Kay, and more specifically thinking about seagrass, seaweed interactions. So digging deeper into what I've learned at Davis, but kind of expanding more, being able to pursue those side projects I started at Davis and building my network. So thank you, CPB. Yeah. Sorry. Okay, yeah. So I've also talked a lot about my research, but I would be remiss if I also didn't get a chance to thank CPB and the Coastal Marine Science Institute for also allowing me to find my voice in service within science. So besides working with faculty and learning how to research or how to do ecology, I've also had the privilege to work in marine science with high schoolers, undergraduates, graduate students, and postdocs at my time here, during my time here. And so in conversations with my mentees in marine science, I learned that as much as folks love marine science, sometimes it can be a little scary and it can feel a little unsure about your future within marine science. So all of these conversations and being able to work with so many different types of people really inspired me to develop a philosophy of service that seals the leaks during key transitions to further retention in marine science. So over the past 6 years, this has culminated in many collaborations with CMSI to build field trip itineraries, building experiential learning for high schoolers out to Bodega Marine Lab, developing 2 classes for our marine and coastal science undergraduate community at Davis, 2 workshops on how to get careers within federal and federal and state resource management for our undergraduates, graduate students, and postdocs within UC Davis more generally, and also working with CMSI and Bodega admin to write onboarding documents on how to get people out at Bodega Marine Lab because it's such a beautiful place. Yeah, so I'm really also, as much as I'm excited to develop more as a researcher after I leave Davis, I'm also excited to continue expanding my service philosophy as a Moore Fellow, to build my philosophy of retention by preparing graduate students for careers in ecology and evolutionary biology more generally. So I'm going to do my acknowledgements. I'm just gonna do them right away. Yeah. So when I applied to Davis, I read my application essay, and the first sentence of my application essay said, "It takes a department to raise a scientist." And I talked about all the labs that I worked in at Georgia Tech. But the longer that I'm in science, the more that I realize that it really takes a village across national schools, UC campuses, as well as people both within academia and outside of academia. So Jay, I'd just like to thank you for being, this is Jay's lab. We talk through sarcasm. That's how we show, that's our words of affirmation. So Jay, I just don't want it to be taken wrong. Jay, thank you for being more hands-off than I wanted initially.
Mission accomplished.
Yes. It turned out to be more hands-on than I could have ever needed. Thank you for letting me find my voice. Yeah, thank you. I've also worked in many labs, and I'm thankful for the labs allowing me to be a barnacle onto them and going to their lab meetings. So specifically, Anya Brown's lab at Davis and Bodega Marine Lab, Jonathan Eisen's lab at Davis. Also, thank you for going birding with me. Kate Laskowski, thank you for teaching me about stats. The Stack Lab, obviously. Maggie Sogan's lab at UC Merced. Thank you, Maggie, for teaching me about chemistry and everyone in your lab. And Peter Dorenstein's lab at UC San Diego. Thank you for teaching me about non-targeted metabolomics. Thank you all for taking me as one of your own and allowing me to join your labs and your lab culture. I'd like to thank my committee members, Anya Brown, Eric Sanford, and in addition to Rick Harbin and Andrew Whitehead. And then also, just quickly, Anne, Anya, Pauline, Eric, Frank, Kate, Maggie, Mark, Martin, Melanie, Moose, Joe, Jonathan, Julia, Rick, Rachel, Susan. When you mentor me, I can feel that you believe in me and that you want me to succeed. That has been invaluable in my development as a scientist. Thank you. Okay. To all the additional educators in my life, thank you. Thank you so much to the support staff of Bodega Marine Lab and UC Davis who made this research run so smooth. And I'd like to thank my family and friends, both from Run Club, from everywhere. Call him boss. And I'm excited for my village to keep expanding in my next step. So thank you. So my dissertation was exploring how microbes contribute to ecological and evolutionary processes within a species. It has been such a joy. For my postdoc research, I'm excited to learn more about how microbes contribute to ecological and evolutionary processes between species. And I'm also excited to continue developing my service philosophy of preparing UCSC graduate students for careers in ecology and evolutionary biology. With that,
You're not out yet. There's questions.
Okay.
You have time for questions.
Peter.
Yeah, fire away.
yes.
That was an amazing talk. Super, super cool. This might be a clarifying question.
Yeah.
But for legacy effects with the
Yes.
microbes versus the plants.
Yeah.
You were looking at the F1 of the plants, but microbes have really, really fast generation time. So how do you think legacy effects would impact a system that has different generation times? And do you think that mismatch could be one of the reasons why?
Yeah, so I'm also sorry if I'm not fully understanding your question, because I'm riding a high, okay? So what I'm talking about is if the amount of provisions you have available across generations impacts your ability to assemble microbial communities. So if you have less provisions available, are you even assembling distinct microbial communities from your surrounding environment?
That was the clarification I did. Yes, thank you, sorry.
Thank you.
There's
Yes.
a question on Zoom. Truman Young said, nice. In the first section, you elegantly demonstrated that variation in microbial communities have impactful consequences. But why do the microbial communities differ in the first place, either in composition or in response?
So that's a good question, because it's also like whenever we do microbial ecology and we're sequencing, we're just taking a snapshot of those microbial communities. So we don't know, like that's the challenge with microbial ecology, is we don't know the before of how those microbes got to the composition that they were in. So it's possible that just standing microbial variation was there from the environment. It's also possible that there were just standing differences in host variation in microbialism was there. So, and differences in microbe-microbe interactions that led to these initial differences in microbial community composition. Yeah, but that's a really good question. And actually Maggie Sovin's group is looking at doing these synthetic microbial communities along with Gina, kind of looking at these dynamics of how microbial communities, like how the microbiome source forms to get more at that answer. So that's a really good question, and the field thinks about this a lot. Yeah. Mhm.
So there are other microbes other than
Yes.
bacteria. Have you ever thought to look at them, or have you thought about whether or not they might have different patterns than what you see with the bacteria?
Yeah, I think about them a lot. Yeah, I think about them. But it's so as I'm learning from Gina, there are challenges with different types of microbes that you're trying to sequence. So for my PhD, I did the path of least resistance, which is the path of B3, B4, 16S. And I'm excited to learn from my lab mates who are right now doing ITS variation to look at protist, labby, like different variation. Yeah, because I've been thinking about these interactions between the bacteria and the wasting disease pathogen, or now learning that there's different fungi and fungal pathogens, and there's just like a whole invisible diversity that's just like, it's inspiring, and I'm excited to learn more from the community and my future collaborators. Yeah, I do think about it a lot. Yes.
Back in your first project, your first chapter, your stressor there was temperature.
Yeah.
Which is cool because it has this kind of effect on everything.
Yeah.
I was sort of started thinking about other kinds of stressors, and in particular like herbivory
Yes.
as something that would maybe really impact the seagrass, but not maybe so much the microbes. I don't know.
Yeah.
Any
Yeah.
thoughts there?
Yeah, so actually, this is a question that the lab has been thinking about a lot, because in land plants, herbivory can induce differences in metabolite, secondary metabolite production, and differences in phylosphere microbial diversity. So how does that relate in marine systems? And so one of my lab mates, who's not with us right now, Nikki Yoon, is really interested in that herbivory aspect. Another aspect that I think about too is shading. So competition between macroalgal species, that also changes the carbon balance, or like where you are putting your chemical communication. So there's so much that we don't know that is so exciting, but Yeah, and there's also research that came out of our lab from Naomi Murray's work on, and Nikki, of the possibility that these invertebrates that graze could be passing wasting disease, or Nikki's first chapter is looking at the dispersal of bacteria with these in fauna. So really, there's a community, both at the macro and micro level. It's really fascinating. Yeah.
Back to the regulators and the conformers.
Mhm.
Did you ever notice, like, among a single site, any sort of geographical distribution of them? For example, certain, like the regulators being more present closer to shore or in deeper water?
Yeah, so this experiment was done in a mesocosm, but that is an excellent point, and I think Anya is thinking about microbial flexibility in the field. Yeah.
Different system.
Different system, yeah, but that's a really good point, and that actually kind of tag teams to what I've been thinking about is like these tidal gradients, because eelgrass is more at the higher tide or at higher tidal elevations. They're experiencing more thermal stress and desiccation stress, or the stress of drying out. So versus plants in the lower tidal, they might be subtidal all the time. They might not be drying out at all. So there could definitely be differences in regulator versus conformer patterns of flexibility within a meadow, just along tidal gradients or across, and in addition to genets or genotypes. Yeah.
Well, let's free Karolina
Yeah.
and thank her and congratulate her once again.